All right. Thank you, everyone. I'm super excited to be here today. For my talk, I will actually be presenting one study that we have recently conducted, looking at the use of network control theory for the personalization of stimulation sites in non-invasive brain stimulation. Sorry.
Can't seem to pass the slides.
Okay, now we can. All right, sorry for that. Anyway, for those in the audience that are not familiar with non-invasive brain stimulation, I'm just going to give you a very short snapshot because it's essential for you to follow the rest of my talk. I'm going to particularly be focusing on transcranial magnetic stimulation, which is a form of non-invasive brain stimulation that allows us to target superficial cortical layers of the cortex. And it does so by means of a series of magnetic pulses that are applied on the individual scalp and allow us to induce secondary electrical currents, which in turn can activate population of neurons. TMS is usually very well tolerated, has minimal side effects, and its mechanism of action are thought to resemble those of neuroplasticity. So this has led to a lot of interest in the use of TMS for therapeutical applications. However, in the recent years, we've also learned that TMS suffers from a very high inter-individual variability, as there are many factors that can influence one's response to TMS, such as, for example, some intrinsic factors like the genetic polymorphism, like BDNF factor, so genetic polymorphism within the brain-derived neurotrophic factor, and some other aspects which are instead more related to the stimulation per se, such as what is going on in our brain at the time of stimulation, so the activity state, but also how and why we choose a certain region to stimulate. And this is the part that I will be focusing more for the rest of my talk. Indeed, TMS has benefited from a very significant technological advancement, and nowadays we do have all the means to co-register in time the position of our participant's head in respect to the coil that is delivering the stimulation, and all of that while referencing to the individual MRI. So we can be sure of targeting a specific cortical folding or cortical gyros that we are interested in targeting. Despite the fact that we can be nowadays very precise, there is still a lot of confusions on how we should actually identify those regions. And I'm bringing to you the example of Alzheimer's disease, in which a series of studies have applied TMS in trying to improve the cognitive functioning of those participants or delay their cognitive decline. And by means of a systematic review that we've conducted, we observed that across studies, the region choice was very different. And you can see here depicted both in the pie chart as well as on the brain template. And what we observed is that generally, yes, all those regions, occupy superficial cortical layers, and then they tend to have a role in functions such as naming or spatial orientation or memory. So those functions that are usually easily and early disrupted in those patients. However, when we look at exactly how they were defined, we observed that at best, if the individual anatomical MRI data were used, but very often, most of the studies actually simply relied on the so-called 10-20 EEG system disposition. So simply targeting an electrode space, assuming that it's overlaying with a given cortical area that we're interested in targeting. And no study whatsoever actually used the individual DTI, fMRI, or PET data to personalize the approach. At the same time, we came across by a paper from Danny Bassett group that started using network control theory to gain a better understanding of invasive electrical stimulation, and in particular, to better understand how the stimulation propagates in the brain. This by means of combining both the individual diffusion-weighted imaging data and the electrical corticography that was recorded in epileptic patients undergoing neurosurgery. And what they observed is that indeed the individual white matter structure was predictive of how influential would have been the stimulation on the brain in terms of how it would shape the induced activity state. And not only, but because they had the ongoing stimulation, they were able to kind of match the predictions made by their model, the network control theory model, to the observed states changes that were induced by means of the real stimulation. So a very nice proof of concept. We hence decided to take up on this approach that they developed, but just applying it at a much larger scale. So instead of focusing just on the neural grid for which they implanted electrodes, to apply this model at the whole brain level, meaning at a whole brain parcellation. And we did so by means of a series of data retrieved by the Human Connectome project, about 400 participants, for which we had both structural and functional data. And our aim in this case was to use the network control theory framework to actually identify what would have been the best stimulation target in the brain of each of our participants if we wanted to induce a desired activity state. For example, the ability to actually activate a given resting state network. And we did so by comparing the effect of stimulation, so the prediction of the stimulation, if that was to be applied to a traditional stimulation site in the literature, as it has been done so far, so one target for everyone, or to a certain degree of individualization, which could have been topologically derived. So, for example, we're taking the nodes within the brain that have the shortest characteristic path length to the nodes that belong to the network that we want to activate. Or we take, for example, the nodes within the brain that have the highest nodal degree to the network that we want to activate, and so on and so forth. Or instead, we simply allow a series of simulations run by network control theory to test all possible regions in the brain and then tell us retrospectively which would have been the optimal one. And we tried to simulate both 1 and 2 regions because nowadays we do have the possibility of using multiple coils, up to 2, to target regions simultaneously and though measure some possible additive effects. Ideally, if a node is efficient for stimulation, then it's a node that allows us to move from an activity state at baseline to a desired activity state, so what we call a target state, and it will do so by bringing our state as close as possible to that target state and in the shortest amount of time. And the model is actually rather simple, and it's a linear model for which we have A, which is our structural connectivity matrix, xt, which is our vector of activity for each one of our nodes. In this case, it was derived based on the resting state fMRI of those participants. Then we have U, which is also a vector that simply represents the shape of our pulse, in this case just a one single pulse, and B, which is a matrix of 1 and 0s that tell us where are we applying the stimulation to, so to which nodes are we actually applying the stimulation. The idea is that if in the literature we are already targeting the best possible nodes in the brain, which are the same for everyone, then the model should just predict those same nodes. But if instead we do assume that differences in the individual structural connectivity will matter in determining how the stimulation will reach the brain, then we should observe some inter-individual differences in the stimulation site. And this was indeed what we observed. So here you see depicted an example for the case in which we want to target the default mode network. And you see here the dots, which represent either the same dot for every participant or a different dot for every participant based on the network control theory prediction, which was the one showing the highest amount of variability. So every dot in the brain represents the optimal stimulation site for one participant, and the size of the dot tells us how many participants out of 400 share the same entry point as their optimal stimulation. We then apply the network control theory framework to map what we call the propagation outreach. So if I apply my stimulus to my optimal site, which other regions are going to be activated in sequential order? And most importantly, do those nodes actually fall within the network that we want to activate? Of course, it's a linear model, as you might have seen, so in trying to make it as plausible as possible, we focused on the very early activation, so the first 10% of nodes activated following the application of the TMS pulse. And what we observed is that network control theory, which is depicted in green, outperformed every other possible stimulation scenario, meaning that it is indeed crucial to personalize the stimulation site in order to be able to successfully reach the target that we want to activate, in this case, the default mode network. And we did so by comparing it also to some null models, so a topological null, so preserving the weight, the degree, the strength distribution, or a spatial null model, so preserving the weight and the relationship between weight and distance between our nodes. And we did so for all the other networks in the brain just to see what could be possible differences in it. And it turned out that it is crucial for every single networks in the brain to actually personalize the stimulation site, with least effect for primary sensory regions. So usually the sensory motor network or the visual network, which makes sense because those networks do not embed associative regions, which are the ones that are most subjective to inter-individual differences. We also tested the specificity, so how many of the nodes that I'm activating actually belong to the target state that I want to activate, and how many nodes belong to other resting state networks. And what we observed is that the specificity of the stimulations was always pretty high, with the exception of 3 networks that you see here highlighted, which when we actually looked further into, they seem to share a significant amount of Y-matter tracts. So that would result in the sense that if we're stimulating the best network control theory derived node for those networks, then there is a higher risk of co-activating structurally connected nodes that they might, however, belong to different resting state networks, as you can see here depicted. We also, because we are working with ACP data, 45 of those participants have undergone a retest acquisition, so 4 to 6 months afterwards. They were undergoing the same ACP protocol twice. And we rerun our model and again observed a very high reliability of the results. So suggesting that even when the states changed based on a different resting state fMRI data and so on and so forth, the predictions were still in favor of the network control theory model. And finally, we decided to challenge ourselves with something that could have been more clinical, and in particular by deriving meta-analytic maps of regions that have been shown to be altered in major depression. So a network of regions that ideally we want to reach with stimulation, which you see depicted in panel B. And again, apply the exact same model, but with having the map on panel B as our target state. Again, finding the same results with network control theory. So the importance of allowing the model to tell us which should be the best entry point. But most importantly, as you see depicted on panel D and E, what we did was then to also overlay what was the propagation outreach predicted by the model, so which nodes would have been activated following the stimulation, and the other underlying fiber tracts. And as you can see, the propagation outreach clearly matches the underlying fiber tracts from departing from the stimulation site, again, network control theory derived. I will conclude my talk in saying that, however, this model suffers from a limitation that we're working at a macro scale level. So we then, I had the pleasure of participating in this perspective paper with many other experts on non-invasive brain stimulation, some also experts in brain modeling. And what we think is going to be the TMS 2.0, so where our efforts should be directed in the future, is actually that of building even more complex models. And in particularly, if you were attending some prior educational courses or other talks, we have been looking over neural mass models. So the idea of being able to actually stimulate the propagation or the activity in the brain at a much lower scale. So looking at the single neural activations. And also, of course, the necessity to actually integrate even more neuroimaging modalities to get more accurate hybrid brain models that would really push forward our level of personalization of stimulation. And with this, I conclude my talk by thanking you for listening and thanking all the collaborators to this project, some of which are present today, and looking forward for your questions afterwards. Thank you very much.